What's up, guys? So today is our first lecture covering protein, function, and enzymes. So this module is going to piggyback directly off the previous module, protein structure. And that's because the structure of a protein is going to directly impact its function. And this is what we refer to as the protein structure function paradigm. And there's 8 broad categories of protein function we're going to talk about today. Now, this classification is not the end-all, be-all. This is just an image that I found somewhere that I really liked. In other sources, you might see them classified a little bit different. It's not too important. The important thing is your textbook doesn't go into too much detail on the wide array of functions of proteins. They really just focus on enzymes. So I wanted to give you a little survey of the other things that proteins can do. So our first class of proteins we're going to talk about are storage proteins. And just like the name implies, these are proteins that act as biological reservoirs. They store things. So a couple examples here, and I don't want you to get lost in the details here. I'm just providing some examples for you to think about how proteins function and how their structures lead to that function. So this first molecule here on the left, or this first protein here on the left, is a protein known as ferritin. So go ahead and pause this video and see if just based on the name, you can come up with a function for the protein ferritin. So that's right, ferritin is a storage protein that stores iron. It stores iron 3+ ions, and it can store a bunch of them. So this is a huge protein. So this part here in purple is a single subunit. It's a single polypeptide chain. And this protein is made up of a quaternary structure of 24 of these. So 24 of these little purple things come together to form this entire structure. So it is a hollow globular protein. And the inside of it can store up to 4,500 iron ions. It's a lot. So this other protein is another interesting one. This is ovalbumin, so chicken egg albumin. So in addition to many other functions, albumin can act as a reservoir for metal ions, but it also kind of stores itself in a sense. It acts as an amino acid source for developing embryos, so it stores amino acids. Our next class of proteins are structural proteins, and that's another protein that you can probably guess from the name what it does. So these are proteins that act as structural components for parts of our body. So a lot of these are going to be fibrous proteins, so proteins that are stiff or rigid. And the best example of that is keratin here. So keratin monomers form these alpha helices, and then 2 alpha helices end up coiling together to produce this coiled coil structure here. And then these coiled coil dimers end up, a lot of them come together to form these fibrils, and that's what's going to make things like hooves or horns or even your hair have this stiffness or rigidity to it. So on the other end of the spectrum, we can have structural proteins that give something elasticity. So these are going to be proteins like elastin or elastin-like proteins. These are going to be disordered protein segments that are covalently cross-linked together. And it's going to form essentially an elastic-like structure where you can stretch it and you can relax it. And the third option for structural proteins is somewhere in between. And that's something that we see with this third protein here, collagen. So collagen, a lot like keratin, has another interesting higher order structure. It forms this triple helical structure. And depending on the amount of biomineralization, collagen can be either hard or it can be very flexible or somewhere in between. Okay, our next class of proteins are hormonal proteins. So I want you to go and pause this video and see if you can give me a definition for a hormone, because we already talked about hormones in the lipids module. So that's right, a hormone is a signaling molecule. Hormones are signaling molecules that regulate the function of distant tissues. So they are produced somewhere else in the body. They're produced in glands, and then they travel through the circulatory system to somewhere distant and regulate certain functionality there. And we already talked about 2 classes of hormones when we covered lipids. We talked about steroid hormones and eicosanoids. And eicosanoids should really have an asterisk next to it here because it's hormone-like. So they do regulate tissue function, but they are produced locally. They're not produced in distant glands. So let's talk about a couple examples of hormonal proteins. And probably the best example is something that you have heard of, insulin. So insulin is going to be the main anabolic hormone in humans. So in response to an increase in our blood glucose levels, it's going to promote the absorption of glucose from the blood into liver fat and skeletal muscle. So in response to a rise in blood glucose levels, it's going to promote the absorption of glucose from the bloodstream to our liver fat and skeletal muscles, where it's going to be converted into glycogen, the glucose storage molecule that we learned about in the carbohydrates module. And we're going to talk a lot more about glycogen when we get to the last module, metabolism. So another cool thing about insulin is that it is a great example of a process known as post-translational modification. So that means once the polypeptide chain has been translated, we have to make some alterations to it before it can be fully functioning. It's not functioning until it goes through a series of post-translational modifications. So the initial polypeptide chain that we get once insulin has been translated is called pre-proinsulin. And it consists of this N-terminal signal sequence, a B chain, a C chain, and an A chain. So that signal sequence is going to guide this nascent insulin molecule to the rough ER. And once it gets there, a protease is going to cleave off the signal sequence. And so we're jumping ahead a little bit here, but a protease is a type of enzyme that cuts up proteins. It hydrolyzes the peptide bonds that connect amino acids together. So once that signal sequence has been cleaved off, we're going to stabilize this structure with 3 disulfide bonds. 2 of them are going to connect the A chain to the B chain, and 1 is going to be solely within the A chain. And we're going to call that structure proinsulin. So then another protease is going to cleave this proinsulin in 2 separate places to cut out this entire C chain here. And once this entire C chain has been cut out, we have a fully functioning, mature insulin molecule. So another good example of a protein hormone would be the opposite of insulin, would be glucagon. So glucagon is the main catabolic hormone in humans. So catabolic meaning breaking larger biomolecules down into smaller components, and anabolism on the other side is building up larger macromolecules from smaller components. So when our blood sugar gets too low, glucagon is going to stimulate the liver to convert the stored glycogen that it has, start cleaving off individual glucoses, and release it to the bloodstream to get that blood glucose level back up. Another good example in us mammals is the hormonal protein prolactin. So prolactin is going to function by stimulating mammary glands to produce milk. Okay, our next class of proteins are contractile proteins. And I've included a video to watch on this that's going to go into a lot more detail. I'm not going to test you over this video, but I would encourage you to watch it. It's very good. And you'll probably go over this stuff a lot more in your anatomy classes as well, and that video should help you out there also. So what I want you to know for this class is the 2 major contractile proteins, myosin, which is also known as the thick filament, and actin, which is also known as the thin filament. And something else I want you to know about actin is that it is also a component of cytoskeletons. So cytoskeletons are part of our cellular architecture. It's this network of interlinking protein filaments. It's not just actin, there are other proteins involved, but they extend all the way from the cell nucleus to the cell membrane. And they function to give shape to our cells. They give them a specific shape, and they give them resistance to deformation. So they kind of protect the cell. So in this sense, actin is kind of a multifunctional protein. So this is where these 8 broad classes break down. So in addition to being the thin filament involved in contractile proteins, you could also argue that actin is a structural protein as well. I would argue that. Okay, our next class are receptor proteins. So receptor proteins are proteins involved in signal transduction. So this is a process where chemical signals are transmitted through a cell, and this ultimately leads to a cellular response. These are usually a lot of steps involved, multiple proteins involved. So the basic way that these complex pathways work is A signal is going to bind to your receptor protein, which is going to cause a change in the conformation of that receptor protein. And that's going to lead to some kind of downstream response. Chances are it's going to be picked up by another protein, which is going to cause another conformational change and another response. So we call this a signal cascade, and it eventually leads to a change in the cell that we need to happen. So what type of changes, what type of responses are we talking about? Well, there's a lot of examples. So one example would be gene regulation. So determining whether a gene should be expressed or how much it should be expressed. Another example would be protein translocation. So kind of like we talked about with insulin, there's going to be receptor proteins involved in getting that pre-pro insulin to the rough ER. These are also involved in cell growth and cell death, so apoptosis, which is programmed cell death. So when a cell is no longer of use or it's become a hazard, like it might have a mutation, which we're going to talk about more in Module 7. So an example of a receptor protein pathway that I really like and will tie in nicely with what we covered in the lipids module is LDL receptors and receptor-mediated endocytosis. So once again, I don't want you to get too lost in the details here. I'm just going to walk you through it because it's just really interesting. I want you to have an appreciation for how these things occur in your body. So the long and short of it is all nucleated cells are going to have these LDL receptors here. And they're clustered in what we call pits, clathrin-coated pits. So clathrin is a protein that when they interact with each other, form these cage-like structures that you see here. So basically what happens is you'll have an ApoB100 protein on an LDL or an ApoE protein on chylomicron remnants or IDLs that have affinity for these LDL receptors. So they'll bind to these LDL receptors. Once that happens, it's going to cause a conformation change in the receptor, which is going to trigger a conformation change in the clathrin proteins coating this pit. It's going to cause them to cave in and form this nano cage structure here. So as this pit falls in on itself, it's going to pinch off from the cell membrane. It's going to form what we call an endosome, which is essentially a small membrane-bound compartment within the cell. And this endosome is going to go deeper into the cell until it merges with another membrane-bound compartment called a lysosome. So a lysosome is an organelle that contains enzymes that will essentially chop up this entire lipoprotein particle and release the free components to the cytoplasm. Okay, our next class of proteins are antibodies, or also called immunoglobulins. So antibodies are Y-shaped proteins, and they're produced by white blood cells, and they're used to neutralize pathogens. So something like a virus or a bacterium that has invaded our system that we've got to do something about. So at the top of the Y on this antibody structure here, we have what we call antigen binding sites. So these are areas that have a specific binding affinity to some kind of protein or sugar sticking off of this pathogen. So the way it works here is these antibodies are secreted, and when they come into contact with a pathogen, more specifically the specific antigen that they have affinity for hanging off of the pathogen, they're going to bind to it like you see here in this little cartoon where you have this green bacterium, this red antigen, probably some kind of glycoprotein hanging off of it. It's going to bind to the top of this Y area of our antibody. And so this bottom, the base of this Y, is going to have affinity for some receptor protein on the cell surface of a white blood cell. And that's going to cause this white blood cell to essentially seek out and destroy this tagged pathogen. So in this video right here, you essentially see this happening. We have a bacterium that has an antibody bound to it, and this neutrophil, a type of white blood cell, is just attracted to it. It's going to seek it out until it binds, and once it binds, it's going to suck that bacterium in a process called phagocytosis, where it essentially digests the bacterium and then spits the remains out. Okay, and our next to last class, our last class for this video, because enzymes, as you're going to see, are kind of special, so we're going to give them their own day in the sun. But this next to last class are transport proteins. And this is another protein that you can probably figure out what it does by the name. They are used to transport stuff, to move something from one part of an organism to another. Most of these, but not all of them, are going to be integral membrane proteins. So proteins that are embedded in a biological membrane. And we're going to subdivide these integral membrane proteins into carrier proteins and protein channels. So this graphic right here shows both a carrier protein and a protein channel. So carrier proteins, these have a specific binding site. So this is like the example you see at the top, where a molecule or an ion has to bind to the protein, which causes a conformation change and opens it up to the other side of the membrane. So it's only open to one side at a time. And these can generally move somewhere between 100 and 1,000 molecules at a time. So the other type, protein channels, don't require a binding site. They're essentially, this tube that's just open. It's open to both sides of the membrane at one time. And since no binding event is required, it can move millions of the right type of ion or molecule at a time through it. So a couple examples of transport proteins. So go ahead and think for a second. Go ahead and pause the video. Can you think of a transport protein that you have already read about in this protein chapter that is not a membrane protein? If you said hemoglobin, I would agree with you. Hemoglobin is a transport protein. It's used to transport oxygen. So let me give you a couple examples of integral membrane transport proteins. So an example of a channel protein would be what we call an aquaporin. So this is a protein. It's going to be open to both sides of the membrane at once, and it specializes in moving water molecules. So we know from the lipids module that water can freely diffuse through a biological membrane. So the purpose of aquaporins is to allow it to happen a lot faster. With these specialized protein channels, we can move water much faster through the membrane than we otherwise would be able to. So an example of a carrier protein would be what we call a sodium-potassium pump. So I'm not going to go into too much detail about that here, but if you're interested, go ahead and click on this link that I have at the bottom of this slide and look at the animation for a sodium-potassium pump. But basically, this is a carrier protein, so it requires a binding event, so it's got specific binding sites for both sodium ions and potassium ions. So this sodium-potassium pump is going to work by transporting 3 sodium ions to the outside of the cell, so out of the cell, for every 2 potassium ions it pumps into the cell. And this is not a favorable process. So if you look at this image right here, if these sodium ions are these little yellow balls, and we're pumping them out of the cell, we are going against the concentration gradient. So that is not favorable. So we have to couple this with the hydrolysis of ATP. And we're going to talk a lot more about that in our metabolism module. Okay, so one last question for you before we finish this up. So in the lipids module, we talked about 3 separate processes to move something across a biological membrane. We talked about simple diffusion, we talked about facilitated diffusion, and we talked about active transport. So go ahead and pause this video and see if you can classify both of these processes here. So classify the movement of water across a cell membrane using aquaporins, and classify using a sodium-potassium pump to pump ions in and out of the cell. Okay, so if you classified the use of an aquaporin as facilitated diffusion, I would agree with you. And if you classified the sodium-potassium pump as active transport, I would agree with you. So they both require the assistance of a protein, but the carrier example also requires an input of energy, the hydrolysis of ATP.